Yeah, we can hear me. Apologies, we've had a few technical issues this evening. So I want to apologise for the start for the delay, but we're in and we're ready to go. So I'll just share my screen. Oh no. Perfect. So yeah, apologies again for the delay, but I'd like to welcome you all along this evening to our webinar titled Updating Lamb Intake Predictions. So this webinar is outlooking a project that's been we've been running for the last year, which has been kindly funded by the University Innovation Fund from the Scottish Funding Council. So we'd like to kind of first start off by thanking the Scottish Funding Council and the University Innovation Fund very much for supporting our studies. So in tonight's webinar, we're just going to start with a little bit of housekeeping. There is ROSA points, so the Register of Sheep Advisors. There's 2 points available for this webinar for any members. So if you would like to collect ROSA points for the webinar this evening, if you contact livestock@scc.co.uk with your membership number, and you can register for your points that way. Also, this webinar is being recorded, so we've if you kind of want to unmute and ask any questions, we'll have an opportunity at the end. If not, please add them into the chat box and we will answer them as we come to the end of the webinar. Or alternatively, I've also put my email in at the end of the webinar, so I'm happy to take any emails as we go through. So just a bit of an intro for tonight's speakers. So tonight we've got myself. So I'm Lorna Shawn. I'm a ruminant nutritionist within the SEC sheep and beef team. So I specialise in sheep and beef nutrition with a particular interest in kind of balances of protein and just kind of balancing rations as closely to requirements as possible, which is where my interest for dry matter intake comes in. We're also joined by Nicola Lamb. So Nicola is a researcher based at the SRUC Hill and Mountain Research Centre at Kirkton, and her specialism has been in sheep, particularly sheep research and genetics. And most recently, she has been looking into methane emissions and lamb efficiency. So she's one of the perfect speakers for tonight. So just to kind of get us kicked off, I wanted to kind of start by going over, what are the benefits of knowing dry matter intake for when we're producing lambs or for stock in general. So one of the first benefits is we can better calculate feed allocation. If we know how much they're going to eat and how much they require in order to get their nutrients, we can predict kind of, particularly at night wintering or finishing lamb grazing scenarios, we can better predict our feed allocations to more efficiently utilise that feed. We can also calculate feed efficiency if we know how much they're eating. So feed efficiency is calculated by kilograms of dry matter going in versus kilograms of meat coming out. So if we know how much we're eating, we can calculate how efficiently they're converting that feed into the final product. We can also better identify when performance is suboptimal. So if we know how much they are meant to be eating, we can then quickly identify if intakes are below what we expect or above what we expect, and actually kind of calculate, is there something going on before we maybe spot a major issue. We also have health. So dry matter intake is a key indicator of health, and often when we find stock are out of health or having a health issue, dry matter intake is one of the first things to drop. They'll stop eating, and I think that's a key thing. If we're monitoring dry matter intake, we can get an early indication of when we maybe have a problem. Ideally going along with this when performance is suboptimal as well. And also, is the diet balanced? If the diet isn't balanced, so it's low in protein or too high in fiber or starch, intakes can be compromised as well. So it's a really good indicator of when if the diet's balanced, or if we've got an imbalance, are we needing to correct that as we go through. So just a quick outline of the project itself. So our Predicting Dry Matter Intakes project came along when we were working with a rationing software package to try and develop a sheep module on it. And when we were doing this, we had a working group and we were discussing different requirements that we were needing to kind of best predict diets. And when we were looking into predicting dry matter intake, there's a common kind of assumption within nutritionists and out with that usually lamb intakes are typically around 4% of body weight. But we delved into kind of, published figures quite closely as well. And when we were delving in, we could see quite clearly that the current dry matter intake predictions, which are based on AFRC, which are actually a technical committee that came together to write the energy and protein requirements of ruminants, which is our current UK guidelines for beef and sheep, energy, protein, and dry matter intake predictions. And it was based on work through the AFRC in 1980. So this is about 40-year-old work. AFRC 1993 is the latest published version of this. And in AFRC 1993, it actually states that in 1990, when they were doing the review, they found these equations to be outdated and gave large differences between predicted and observed intakes. We've not seen any updated intakes since then. So we kind of recognize there's a large gap in knowledge to try and predict dry matter intakes during this stage. And this is what brought about our project. So as part of the project, we've pulled together a working group, which consisted of SEC specialists. So we've got Poppy Frater and Kirsten Williams, who are both sheep specialists. We have myself and Karen Stewart, who are both nutritionists. We have SRUC researchers, Nicola Lamb and Anne McLaren, who are both sheep researchers based at Kirkton. And we've also had 2 independent sheep nutritionists, Leslie Stubbings and Kate Phillips. So we've had a really nice rounded group to discuss the data and discuss what our next steps and what we want to get out to get a more accurate prediction of dry matter intake. And our main aim in the project was to disseminate results from current intake studies undertaken, to update lamb intake predictions to better replicate dry matter intake predictions for the modern lamb. So we've been looking through a lot of data to try and identify what, how can we better replicate our predictions for the modern lamb, and how does actual intakes compare to what these predicted intakes are as well. So what we've done so far in the trial is we've started off with looking at pulling together the working group so we can look at the data, we can disseminate the data, and we've discussed results and next steps. And after every meeting, we've kind of had next steps and we've pulled those steps together and we've looked at something else to try and get as wide a picture as possible. And that's what we're hoping to show you during tonight's webinar. And we've also looked in great detail at the intake data from existing SRUC studies, primarily grass to gas and concentrate studies. So that moves me on quite nicely to Nicola, who's now going to take you through a bit about a background about the grass to gas and concentrate studies that we've used for the trial, and also about the feed intake bins that we've used at Kirkton to look at data. I'll unshare my screen, so.
Okay. Thanks, Lorna. I'll just swap over to mine. Hopefully that will work smoothly. Can you see the PowerPoint okay? Yeah. Okay. So as Lorna said, we had some results from trials that we've been doing as part of the research group, looking at feed intake and feed efficiency at the individual animal level for sheep. And we thought that some of that data could feed into this project. So this is our setup that we've got for measuring individual feed intake. And this is set up at the Hill and Mountain Research Centre that both Anne and I work at up near Crianlarich. So it's a series of forage bins, concentrate crates and water crates. So we can record individual animals' intake of forage, concentrates and water using this kit. And it works with the electronic ID tags in the rears. So every time an animal goes into one of the bits of equipment, it reads their tag and we get a row on a spreadsheet on the computer in the control room that shows us which animal's eating, the time it entered the feeding station, and the weight of feed or water that it consumed during that time, and the duration that it took for that meal or visit. So the way our system works, we usually give them ad lib access to the feeders, and for the trials that I'm going to describe tonight, we left the doors down, so you can have it where they're only allowed to access certain feeders. But for these trials, we gave them access to all the 16 forage bins. So these are the green bins that you can see in the picture here. So we set it up as one big pen. So we have a group of lambs in there after weaning. So we usually have up to about 125 lambs in the pen. They can access any of these 16 bins ad lib. And the way we work it, we usually have a 2-week training period where we train the animals to use the feeders, and we can monitor the data to check that they're all using the feeders and eating enough feed. And then we have a 6-week feed intake trial where we take very detailed information on every meal that the animal eats and also record their live weights. So for most of the trials that I'm going to talk about, we used grass nuts in the roughage troughs. I'll talk about one trial at the end where we used concentrate finishing pellets, but again, we just used them in these green forage troughs, so they had ad lib access to the 16 forage bins. And the ratio of bins to lambs, there's been quite a lot of previous research done on different types of systems, but they found that a ratio of about 9 to 11 animals per bin gave a good representation of how the animals feed in a kind of group situation. So we wanted to use some of the data that we'd collected in previous research trials to feed into what Lorna was mentioning for these lamb feed intake calculations for use in modelling. These have been collected during previous research trials, so the kind of downside was that these trials hadn't really been designed for that with that purpose in mind. Most of them were looking at collecting data that could potentially be used in breeding programs. But we did have data from different breeds and crosses from the trials and from different sexes. So we had some females, castrates, and entire male lambs that had gone through the system. Mainly on the standardized grass nut diet, and that's really because we wanted to compare them on a forage-based diet and with a more standardized diet. So the pelleted grass nuts were used for most of those trials, although as I mentioned, there was one where we used concentrate finishing pellets. And we wanted to look at the potential of using this data to update these lamb feed intake models. So just a bit of background on the projects that we collected the data on. The first was the Grass to Gas project. So this is an international ERANET project. So we were one of 7 countries that were involved in the project. And it was really concentrating on feed intake and methane emissions from sheep. So in the trial that we did at SRUC, we did it over 2 summers, so 2021-22, and we used Texel Cross mule finishing lambs. So in total, we had 481 across the 2 years. And they were a mix of females and castrates, about half and half. And they were sired by performance recorded Texel sires with a range of ABVs. And half of the lambs, roughly, were recorded indoors through the feed intake equipment and were fed grass nuts. So that was about 120 of these each year across the 2 years. And we weighed the lambs every week, and for every ton bag of grass nuts that we used, we got the sample of the feed analysed, which I'll show you some results from later on. As part of the wider trial, we also CT scanned the lambs and ultrasound scanned them at the start and end of the 6-week feeding trial to look at changes in body composition for use in our efficiency calculations, and also to look at CT measured rumen volume, and this was as a potential predictor of methane emissions from individual animals. And the aim was to look at the factors affecting feed efficiency in these lambs that were measured indoors. And then we wanted to kind of see if the lambs that were finishing indoors were performing differently to their relatives that we finished outdoors on grass, because the indoor situation was really not how these animals would normally be finished in the UK. So we split pairs of twins, and for each animal that was finished indoors, we put their twin outdoors on pasture. So we had about the same number, 242 lambs that were grazed on pasture, and again they were split across the 2 years. And from these lambs we recorded the grazing offtake, and Lorna will show you some of the calculations we did with that. Again, we measured their growth and body composition, and from a subsample of them we measured the rumen volume using CT scanning as well. So just some results from that, just to look at feed intake or feed efficiency from a kind of genetic side of things, which is where that project was really focused. We had different sires represented across the 2 years. So each of those graphs is from one of the 2 years. And this is the average dry matter intake and average daily live weight gain from the lambs that were produced from each of the sires. So you can see the bars, the red bars on the 2021 graph and the orange bars in 2022, they're showing the average dry matter intake of the lambs from each sire. So you can see there's quite a lot of variation in those averages between the sires. And then the little diamonds that are above the bars or close to the bars show the average daily live weight gain for those same lambs. So you can see that some of the sires had relatively high dry matter intake in their lambs, but relatively low average daily live weight gain in sire E there on the left-hand graph, for example, whereas some others looked a lot more efficient. So sire H on the right-hand graph there had lower dry matter intake in his progeny and a higher average daily live weight gain than some of the other sires. We also wanted to look a bit at how the lambs finished indoors differed to those finished outdoors, and we saw that there was significant differences in the performance due to the system or diet. And actually those that were finished indoors had higher growth rates and higher measurements of our carcass fat and muscle than the animals that had been finished outdoors. And particularly in these 2 years, we had very dry summers, and we did struggle a bit with the grass quality outdoors. So those that have been finished indoors actually perform better. And we also found there was significant differences in performance due to the sire. But then when we compared those sire differences in the indoor versus the outdoor system, we found that the same sires ranked similarly in each system. So the ones whose lambs grew the best indoors also grew the best outdoors, even though there were differences between the systems. And the same was true for the carcass traits and also for this measure of rumen volume that we can take with CT scanning. And the graph there just shows an example with the rumen volume. So the bars, there's a red and a blue bar for each sire. The red bar is indoors and the blue bar is outdoors. So the outdoor lambs tended to have bigger rumen volumes after they'd been outside for the 8 weeks, whereas those that had been inside on the grass nuts had smaller rumens. But we can see that the way the sires ranked was quite similar between the two different systems, and we saw the same for growth and carcass traits as well. Although the order was swapped around, so growth and carcass traits with the indoor animals performing better. So this suggests that genetic differences we see between sires persisted when their lambs were being measured indoors or outdoors. So it gives us some confidence that what we're measuring in an indoor system when we're looking at feed efficiency is similar to what we would see if the animals were finished on grazing. And this has been shown in the literature as well, that measuring feed efficiency, particularly on a forage-based diet indoors, reflects feed efficiency measured at pasture. So the other data set that we had available was using the same equipment, but measuring some of the hill sheep on our Scottish Government funded research project at Kirkton. So we've had a long-running selection line of high-index Scottish blackface sheep that have been selected on maternal traits and lamb production traits in a selection index. And for the last few years, we've been crossing the blackface with the Clin flock that we had at Kirkton as well. So we have now a mixture of Scottish blackface and Clin crossbred animals. And we have both crosses, so we have blackface sire with Clin dams and the other way around as well, because we started off with a ewe flock of each of the two breeds. So for a couple of decades now, we've been selecting these Scottish blackface and also the Clin flock for production and health and welfare. And now we're adding in new traits that we're recording to do with resilience, efficiency, and environmental impact. So one of the traits that we're adding on to our recording is the feed intake and feed efficiency. So in a very similar way to what we were doing with the grass to gas lambs, we're measuring individual feed intake on a sample of 120 weaned entire ram lambs this time per year. And these are a sample across the different sires that we have in the flock each year. And we're taking our weekly live weights on these lambs in the same way over that 2-week training period and 6-week feed recording trial. And you can see some of the first results here from the first couple of years. The main aim isn't to really compare between breeds or crosses, but really to mix up the genes from the two breeds and see which come out best in terms of efficiency and also these other traits that I was talking about. But we have seen preliminary differences between the different crossbred lambs and the pure Scottish blackface lambs so far. And again, we're CT scanning them before and after the 6-week feeding trial to look at carcass composition changes and rumen volume. So we've had about 100 per year that we've had full records on through the feed intake recording, and we're just about to finish the 3rd year of that data collection. So the 2023 lambs are in the feed intake recording equipment at the moment. So we have our pure Scottish blackface high index line, and then we have at the moment different crosses. So we have the blackface cross Clin the other way around, and then the second cross. And we've just finished this, we're finishing the 2nd year of recording in them. So again, they were fed grass nuts to try and standardise the diet, and we analysed a sample from each of the bags of grass nuts. And this is the sort of feed analysis that we get when we send the sample of grass nuts to the SAC lab. So when I was looking at the feed efficiency, I was only really homing in on the dry matter content from the results that I get, but Lorna, being a nutritionist, looked at these results in a lot more detail and came up with with other ways to analyse the data as well. I mentioned that we had a trial using concentrate finishing pellets. So this was, we used the equipment to finish the hill lambs. Again, they were entire male lambs from the hill flock in 2018, 19 and 20. And in each of these years, we had about 100 pure Scottish Blackface and pure Clin ram lambs. So that was before we started the crossing programme. And they were either finished in a mixed breed group in the first 2 years, or in the second year we split the Blackies from the Clins. And again, we recorded them using the feed intake equipment, but we used concentrate finishing pellets, but just in those green forage bins with AdLib access to all the lambs. And again, we did some feed analysis on the finisher pellets. As I mentioned, these trials weren't really designed to collect the sort of information that would be most useful for Lorna's calculations. So this is a less clean data set, so the protocols were less standardized, as well as giving them the finishing pellets. We also gave them straw that they could eat AdLib, and that wasn't recorded. So we weren't really recording total intake during these finishing periods. We were really just looking at recording the amount of concentrates that they were eating. Also, one of the issues with this data set was that the lambs were only in the equipment until they were ready for slaughter. So we drew lambs every couple of weeks that were ready for slaughter. So that meant the stocking density went down over time in the pen, and the test duration changed as the lambs finished. But at the time, we were only really using the equipment as a kind of look-see to see what data we could collect and whether we could see any differences between the amounts of finishing pellets that the different lambs were eating. So I think that's all I'm really going to say on the data sets, and then I'll hand back over to Lorna to explain how she used that for her analysis.
Right, thanks very much, Nicola. Oh, that's not sharing the right. Screen share. Oh no, that's not. Visuals. Oh, there we go. Perfect. So like we had the kind of data set from Nicola where we had quite a lot of data from different lambs and different trials on individual feed intakes. So what we've done, we've got the actual daily dry matter intake of these lambs. So we've then went back and had a look versus actual, and then kind of modeled it into the current predictions. So these are the current predictions from AFRC. So AFRC predictions currently splits diets into 3 different sections. So we have coarse diets, which are diets containing long or chopped forage. And for that, this here is the sum here. I don't expect everyone to remember this equation, but this is the equation that's used where QM is actually a measure of diet quality. So it's your gross energy divided by your metabolizable energy, which is your energy used by the lamb. And W in this equation is weight. So we can see this calculation is taking account forage quality, but also the weight of the lamb. We also have a prediction for fine diets, which is looking at diets on more a concentrate basis, or also ground and pelleted forage, such as your grass nuts, which we were looking at in the grass-to-gas trials and the feed intake trials, and also the concentrates as well. So for this, we're also taking into account diet quality and weight with a slightly different equation. And when I worked this out for an average lamb with an average quality diet, we were looking at about 2.4% for our coarse diets and 3.9% of body weight for our fine diets. So you can see for kind of concentrate grass intakes, that kind of 4% figure that we've been widely using is in line with that prediction from AFRC. And if we were just feeding silage only, there is a diet for silage dry, calculation for silage dry matter intake, and that doesn't take account quality. So that pretty much sits around 2% of body weight for silage only, but we would need to account for diet quality as well. So your kind of coarse diet is probably a bit more accurate to ensure we're putting that in at the moment. And this is just a bit of a key. So our BW is our body weight in this situation as well. So these are the current predictions from AFRC. So we also had a look at what was in rationing software as well, using the software package that we use ourselves. And this was using around that kind of 4%, but adjusting for ration quality throughout as well. So this graph here shows the data from our grass-to-gas trial. So this was grass pellet intakes using your Texel cross mule lambs, sired by Texel. And this shows dry matter intake as a percentage body weight. So all of our green lines here are the actual average daily dry matter intake for that trial. And the light lines are 2021, dark lines are 2022. And if we look at the blue lines, that's our AFRC predictions. And the orangey yellow lines were the predictions from the software package if we put in the quality of the ration in as well. So for every kind of intake we had in terms of average daily dry matter intake, I've also calculated using the predictions and used the analysis of the grass pellets to get an accurate prediction from AFRC and also the software package. And we can see when we work through that, we were finding in the grass-to-gas trials that we were getting that kind of average daily dry matter intake. So this was the actual dry matter intake of the lambs. It was sitting around that 4% on average, quite consistently. We were changing in between about kind of just above the like 4.5% going down to the 4% in 2021, and we saw a bit more of a dip towards just kind of 3.4% ish in 2022, but largely on average we were sitting around that 4%, which gives us quite good confidence that when we're predicting 4% of dry matter intakes for these types of diets that we're quite close. But what it did highlight was AFRC pretty much consistently sits under on predictions, and the ration software was slightly over predicting intake. So it definitely did highlight that we have a good case to be looking at predicting dry matter intakes more accurately and looking at reviewing these equations to get more accurate predictions in future through software, but also in published data. So I've included this table just as a bit of a snapshot. So this was 2021 data, just showing the live weight of lambs. So we can just see what kind of weight of lambs we were working with. So we were starting with about 36, well, kind of almost 37 kilo lambs, working up to about 47 kilo lambs. So they were being taken fairly heavy. And just to show you the daily live weight gains we were gaining on the grass pellets as well. There was a wee bit of fluctuation as we go through, with not much of a difference in terms of dry matter intake, which was quite interesting. But this is just kind of a snapshot of some of the data we were looking at without too much detail. There's a lot more, and it gets a lot more complicated when you start looking at the spreadsheet. So This is, we've done the same for the Kirkton feed intake data. So this was the entire ram lambs. And again, we're seeing a fairly similar pattern. There's a bit more fluctuation, specifically with our software predictions in this case. As kind of Nicola mentioned, these trials weren't formulated to be feed intake trials exactly. And when we were looking at data, the most important factor for the data for these trials was the dry matter of the grass nuts. So we weren't really looking at energy and protein as much. When I did go through all of that data, I did find that some of them didn't have predicted energies already that was analysed due to what had been predicted. So I've had to use the modified acid detergent fibre to predict energy value from that data, because we did have that information available. And we can predict energy from that. So that's where we've maybe seen a little bit of a difference in here. But largely, we can see that, again, we're seeing AFRC is under predicting. Average dry matter intakes were very much around the 4% again, which was very interesting when we were comparing that to grass to gas, that was looking at females and castrated males, and more kind of Texel cross lambs versus the Kirkton feed intake data that was looking at blackface and blackface cross lambs, and they were all entire rams as well. So we weren't actually seeing much difference between breeds and sex as well, which was quite interesting through this data. And we were largely, again, sitting around actual dry matter intake of around 4%, with in general software over predicting and AFRC under predicting, which was a trend we kind of saw throughout. So we also looked at concentrate, and Nicola mentioned this was definitely not designed as an intake trial, and we were also providing straw, which wasn't measured. And then also we had fluctuations in terms of lambs being drawn out and different group size changes as we go. So this data was a lot more erratic. It was quite interesting when we graphed it out that it's actually ended up mirroring each other across the 2 years. I think this has probably been merely a coincidence, and it would be interesting to maybe follow this through to see what would come in following years. But largely we found intakes to be kind of lower than that 4%. In 2022, we found them to be quite a bit higher than our 2021 data, which we saw a bit of a fluctuate more down to the kind of 2.2%, which had gone. So in general, on average, our concentrate intakes were lower. However, we weren't taking account of straw. We had group size changes. We would need a lot more data to validate this before we looked at doing an actual intake prediction for concentrate. So this was one thing we discussed in the working group, is, you know, what data is usable, what would we be confident with, and what do we need a bit more information for. We also looked at grazing intakes. So Nicola mentioned we used grazing, which was actually predicted using cages. So the cages actually showed our grass growth, and then we were measuring residuals weekly for the grazing data. So it was basically a difference between grazed and ungrazed biomass. We used 6 cages in the paddock to show any, like the ungrazed biomass, and then there was also dry matter content of pooled wet samples taken as well, measured weekly. Some of the paddocks were quite big and uneven, so this is maybe accounting for some of the differences. And it was, if we look back at kind of 2021, 2022 summers, particularly 2022, I think we'll all remember it was rather dry, and we were definitely struggling in terms of grass growth during the point of this trial as well, which might have affected our data. What it has highlighted is the difficulties of gaining grazing data, but also the value of having grazing data for lambs. If we can calculate how much a lamb's eating in terms of when we're putting a lamb in, we can better calculate our moves, and we can better calculate if we're actually meeting requirements. Also kind of coupled with regular weighing, just to check that they were still gaining. but we can get a lot more information by knowing, what are they eating at grazing and what's the quality of grazing in front of them. However, it's a very, very hard thing to predict with a lot of variables. So we had a look at a lot of this data, and our next step in the working group was to actually assess is what is missing from this data. What's the missing piece of this puzzle that we need to actually confidently add in new prediction and put a kind of new dry matter intake prediction equation forward. So I think our first step was our real intakes of grazing. There's not much data on this, and it's very, very hard to do on an individual basis. So we have to do it on a group basis, but we're also got a lot of variabilities in measuring. We also have a lot of different types of grazing, and we need to kind of assess whether there's differences there also. Also, differences between breeds. At the moment, we have had different breeds in the trials and we haven't seen much differences, but more information would allow us to quantify if there definitely is no differences. I think breaking it down into percentage of body weight is a good way to do it because it standardizes the figure and doesn't take into account different body weights of lambs, obviously. Also differences between sex, again, similar to breeds, we have looked at it. We have some data to suggest there's not much differences. However, other data would suggest with castrates versus non-castrates and ewe lambs that there could be differences, especially as they mature, we see a few more differences. So it'd be quite nice to quantify at what stage does this change. And also, we've seen a lot of differences in the last 40 years since these predictions have been made and written. So we're now up to about 90 different breeds of sheep, which has actually increased quite dramatically over the last wee while. We also have a lot of different feeds available to us. We're seeing kind of a lot more kind of clover coming into swards. We're seeing mixed species swards. A lot more different species of grazing coming in. We're seeing a lot more outwintering systems, kale rape hybrids, stubble turnips. We're also seeing winter graze cereals. So assessing kind of dry matter intake of these crops and being able to predict that better going forward would definitely assess us and help us to better quantify how much we're feeding, that we're getting the diet right, and we're being as efficient as possible when we're providing this, when we're providing diets to lambs. So we've had a look, and part of the project is to actually produce a fact sheet on calculating dry matter intake. But there is a few things we can do on a farm basis, and obviously this is looking at groups of animals, as on a farm basis to do individual dry matter intakes is very, very difficult without sophisticated feed bins or kind of other methods of predicting dry matter intake would be using kind of markers, so things like N-alkanes and stuff, which are very difficult to replicate on a farm-based level. So these are just kind of ways that we can measure dry matter and also predict dry matter intake on a farm basis for kind of groups of animals. So, starting with grazing, we've got two options to measure our dry matter availability. We've got quadrats and plate meters. So obviously with a quadrat, it's placed in the area that's representative of the amount of grass in the paddock. Or we can do a couple of different areas and then average it across. We always want to knock the water off because we don't want to be getting that extra water as well. Because we're relying on weight in this stage as well, we want to make sure we're knocking any excess water off. And then we're cutting the grass to what our grazing residual would be. So obviously that would be different depending on whether we're set stocking or rotationally grazing, depending what system we're in. But if we were kind of set stocking, we're kind of looking at that kind of 4 centimeters sort of level. And then calculating dry matter in the paddock. So kind of weight of grass times the dry matter of the grass divided by the kilograms of dry matter per hectare in the divided by about 40,000 to get our kilograms dry matter hectare in the paddock. So there is a way we can predict it using kind of quadrats, and we can do very, very similar with plate meters. If we know this information, so we know how much dry matter is in the paddock, and we can then calculate how long it actually took to reach our grazing residual with how many lambs to get a rough idea of what that dry matter intake would be. If we really wanted to do it a bit more exact, using kind of something like a cage in a couple of spots of the field, representative spots of kind of where grass growth would be typical. We can also calculate how much grass growth we're getting as well, so to take into account growth in that calculation. If we're looking at forage crops, again, using kind of quadrats, we've got to mention that the methods differ for like leafy brassicas and broadcast root crops versus root crops that have been planted in drills. There is a really, really good indication on how to predict dry matter intake of a forage crop in the outwintering guide. So I've included a link to the outwintering guide here. And the webinar is being recorded, so that link will be available in the recording if you want to go back and write it down. And then if we were feeding kind of concentrate, and a quick example for a concentrate, if we are filling a hopper to full with 6 25 kilo bags every 2 days, feeding 100 lambs, we're feeding 150 kilos a day, dividing that by 2 days at 75 kilos. 75 kilos over 100 lambs is 0.75 kilos a day. If we then times that by our dry matter, we're feeding 0.645 kilos dry matter a day. And that was just a really quick example for calculating how much a group of lambs are eating on average on a concentrate basis. We can very much do the same with forage if we know the dry matter content of the forage, and that's one of the most difficult ones with the variability of dry matter of forage. It changes depending on the forage and depending on time of cutting. If we're looking at silages, we can get a very wide range, whereas if we're looking at drier feeds such as kind of concentrates, grass nuts, we're typically looking at that kind of 86 to 88% dry matter on average. And just a quick reminder that our fresh weight times our dry matter equals our kind of total dry matter availability. So that's the kind of easiest way to calculate it out. So after looking at all this data, and we've went through the data in the webinar today, just kind of highlighting our next steps of the project. So our main next step in the project is pulling together a fact sheet on measuring dry matter intake, which is going to feature a standard approach, but also to be more kind of practical to farmers and kind of better predict it at a farm level. And then obviously we do have the gold standard of predicting dry matter intake in terms of individual feed intake bins. We can also use kind of marker-based studies. However, these very much suit a research-based focus rather than on farm. We're also keen to gather data. We have contacted other research organisations and also not just in the UK but abroad also, to see, is there more data on intakes out there that have been qualified that we can use to quantify the data we have from the studies that we've used from Kirkton. And then we're also, keen to just hear information as nutritionists. If we can better calculate how much of a lamb's eating, we can more accurately predict what our energy and protein supply is, and then therefore pull that back to performance and get more accurate rationing. And that's part of the reason why we want to look at dry matter intake. The more knowledge we have, the more accurate we can tailor rations to that animal. So in summary of what we've kind of discussed this evening, on a dried forage diet using grass nuts, actual dry matter intake was very much around 4% of body weight. What we found that current published predictions very much underestimate dry matter intake, and at the moment calculations and software programs are slightly over predicting, maybe not to the same degree that we were seeing in terms of the under prediction from AFRC. Also, in terms of other feedstuffs, very much more data is required before we can accurately predict intakes and come up with a new equation. But it's something we'd be very, very keen to kind of keep going forward with. We've made a good step. We've assessed that there is a need for a new equation and a need for better quantifying dry matter intakes, but we very much need more data in order to actually get that as accurate as possible, and to mimic actual situations to get rations as consistent as we can. And then also we've seen that kind of current silage predictions, and silage was one thing that we didn't look at through this study so far, was around 2% dry matter intake for just the silage only, but this calculation didn't account for forage quality. We would expect to see higher intakes on a higher quality forage, and it would be good to kind of quantify that a bit more. We have a bit of data on the ewe side in terms of forage intakes is in relation to total intakes, and we'd really like to quantify this for lambs also. So thank you very much for listening, and for all of you that stuck around with us when we're having a few technical issues at the start, and can only apologise for that again. And if you've got any questions, I'm sure myself and Nicola would be happy to answer. We do have a couple of questions in the chat as well. Just stop sharing my screen slightly. Perfect. So there is a question asking if it's possible to zero graze 35 to 47 kilo lambs. So looking at a zero grazing approach for lambs. I think it would be possible to zero graze. I think it'd be a difficult thing to kind of calculate how much we're putting in front of them. And we've also got to kind of make sure with a zero grazing system, we've got to be collecting grass every single day, keeping it fresh. There's a lot of work involved in it. So although it would be possible to do that on an indoor system, yeah, there is a lot of work involved in that system. I don't know if you'd have anything.
Yeah, I think one of the project partners in Grass to Gas has a similar system, the biocontrol feed bins, and they were doing some zero grazing, but they have it set up very differently. So it's just a couple of animals in a small pen with access to one bin each and much smaller numbers. So I think they only had maybe 30 pens or something. So they were managing to keep up with the demands, but I think in our system with 120 lambs, it'd be quite tough to keep it going. And the reason that we've gone with grass nuts previously is because we're trying to keep the diet as homogeneous as possible to look at any differences, mainly for looking at ways to incorporate it into a breeding program. Whereas a zero grazing would give you more accurate information on that type of diet, but maybe not so good for the standardized diet for the breeding programs.
Yep, perfect. That's another question came in. We've got a question asking which brand of grass nuts was used.
No, I can't remember. I could look it up for you. So it's just big ton bags that we get from, and it's the same supplier we've had each year. If you give me a minute, if you move on, I'll look up my emails and see if I can find out where they came from.
No problem. Is there any other questions? So there is a question here about grass nuts fed with straw or hay as long forage. In the grass to gas trial, the grass nuts were just fed as a sole feed. Similar to grass, they've already got that kind of functional fibre source in there with a good level of energy and protein. So the grass nuts were just fed on their own as kind of one consistent feed. That was one of the benefits of using the grass nuts for the grass to gas trial, was keeping one consistent feed. We weren't having to supplement an additional forage that we would have to if we were using a concentrate feed, for example.
Sorry, I can't find the grass nut email off the top of my head. Farmers. There we go.
Perfect.
From farmers. Yeah, it's not a cheap feed to feed either. And plus we have to bed them on sawdust so that they've not got any bedding they can eat, which is quite expensive as well. So it's an expensive business to keep them on the grass nuts and the sawdust for 8 weeks. But yeah, it gives good results.
Perfect. Yeah, definitely did for the study. Well, I think that's all the questions. But if anyone does have any further questions, please do not hesitate to email me on lorna.shaw@sec.co.uk. And thank you again for all attending. Hope you all found it interesting. And yeah, hope you all have a good evening.